name is Davide Dulcis, and I am a faculty of psychiatry here in the Department of Psychiatry at UCSD. I was interested in finding ways to stimulate the brain using sensory modalities, and light is one of the ways we stimulate the brain. So by chance we played with photoperiods and we looked in the brain, and we found that some changes were occurring in the hypothalamus. Neurotransmitters have been thought to be immutable, like invariant, and I found that actually this is not true, and that activity can change the expression not just of any other genes in the cell, but also neurotransmitters. One of my students that is kind of feeling blue one day, I say go under the sun for a nice walk, and you will see that just soaking some light will make you feel better. So it's the effect of light on mood, right? And that was the key component that I wanted to understand. We always welcome new undergraduate. One of them is studying the effect of electrical stimulation in the brain and finding where neurons in the brain change their neurotransmitters. And so one of the tasks is to use stereology. This is a program that is used to count neurons. And so they have to identify nuclei, and then nuclei like this one. This is a beautiful image that a postdoc actually generated. And it shows how many cells display and express different transmitters in the suprachiasmatic nucleus, which is the key regulator for circadian rhythms. Now that we have a lot of genetic tools, I think it's time to revisit a bit the way we analyze the circadian clocks. It would allow us to study not just their physiology, but what's the effect of, let's say, a knockdown or knockout of a gene, circadian clock gene, on the function of specific classes of neurons.